the lipid bilayer, that is the plasma membrane of cells, and it has in the center a hydrophobic nonpolar part. And what happens, because there are these molecules, there are the phospholipids, the glycolipids, and the cholesterol. And they have all something with them which makes them polar at one side and non-polar at the other one. They are the phospholipids. They have a phosphor head. That phosphor group head is polar. And the polar phosphor head group head of the phospholipids is hydrophilic. It is polar and hydrophilic. It loves the water. So it turns to the watery portion on the outside in the extracellular fluid, and a bunch of other ones of them are turning to the inside to the cytosol, the watery fluid part of the cell inside of the cell. And so they're just all there, and we're turning around, all with the head facing the phosphor head, the phospholipids, the watery part on both sides. And then they have just then the tail to the inside, the two fatty acid tail, which are hydrophobic, they don't like the water, they turn away, like magnets turning around. And then in the center there is now this hydrophobic, the tails, the fatty acids, they don't like water, hydrophobic non-polar region. And these phospholipids, they are 75% of all these lipids there. And then the cholesterol, they are 20%. And they have an OH alcohol group at their head. And that forms hydrogen bonds with the polar phosphor heads of the phospholipids. So these cholesterol things are also going with the head, with their OH alcohol group head to the heads of the phospholipids. And they have also a tail with steroid rings and a hydrocarbon thing. And then the last 5%, these are glycolipids. They have a sugar carbohydrate attached, and that is doing the same. It's going also there to that. And that way the lipid bilayer is formed. The glycoproteins, the glycolipids, they're going only to the outside. They're only in the outside membrane, and that is why outside layer. And that is why the lipid bilayer is asymmetric, because, for example, the glycolipids are just at the outside. And that is according to Gerard-Tugan-Sender-Grovski's Principles of Anatomy and Physiology book on the pages 62 and 63. But this is now substitute for medical knowledge and also now medical advice. Hi and welcome. I'm trying to learn cytology. Today the lipid bilayer, and after we've done with it, we're gonna do tiptoe yoga. And if you participate in the tiptoe yoga, then it's 30 hours. The lipid bilayer, the basic framework. Let's see first up again. The plasma membrane. The plasma membrane is a flexible yet sturdy barrier that surrounds and contains cytoplasm of the cell. Okay, there's the plasma membrane, cytoplasm consisting of the cytosol, the fluid part, and the organelles, these little ships inside of its cytosol, the submarines, and then the nuclei, the nucleus, with the DNA or the genes in it. So the plasma membrane is a flexible yet sturdy barrier that surrounds and contains the cytoplasm of a cell. The plasma membrane is just the outside wall. The fluid mosaic model describes its structure. Do we mean that here? Plasma membrane depicting the fluid mosaic arrangements of lipids and proteins. The molecular arrangement of the plasma membrane resembles an ever-moving sea of lipids that contains a mosaic of many different proteins. I don't understand what they mean with mosaic. Yeah, they know how they look, but they don't really understand. They just mean there are a lot of them, but it doesn't make sense. A mosaic is fixed or whatever. It's an ever-moving sea of lipids. It's basically like if you see a million water bottles on the ocean or something. They are all just there, but they move. They can move from side to side. Which was here written somewhere. They can move to the other side. The proteins may float freely like icebergs, be moored at specific locations, or be moved through the lipid sea. I don't know what moored means. Whatever, they can be just there like icebergs, freely floating. They can be fixed at a location, or they can completely go through it. Whatever. The lipid act as a barrier to the entry or exit of charged or polar substances, while some of the proteins in the plasma membrane act as gatekeepers that regulate the traffic of polar molecules and ions into and out of a cell. Some molecules and ions are regulated which come in and which come out, and it has a lot to do. We read it all, but no, we're going to be repeating it because we try to prepare our presentation trying to say what is the plasma membrane of lipid bilayer or something. No, and it's about whether they are positive or negative or specific things and they're fitting in or not in. The positive one fits to the outside or not, or it goes in or not, because the lipid bilayer is organized that they are protruding with a positive end or a negative end or something, and now something coming with a positive end can go in, other thing not, and that we have the molecules which have all specific structures, almost like a key actually looking if it or they don't fit and go through or not. So the plasma membrane is looking that only the right substances in the right amount come through. The plasma membrane of typical body cells are about a 50-50 mix of weight of proteins and lipids that are held together by non-covalent forces such as hydrogen bonds. But because membrane proteins are larger and more massive than membrane lipids, there are about 50 lipid molecules for each protein molecule. So the proteins and the lipids in the plasma membrane, which is the wall of cells, have the same mass. But there are 50 times as much lipid molecules as protein molecules. That's made of sense. The lipid bilayer. The basic framework of the plasma membrane is the lipid bilayer, two back-to-back layers made up of 3 types of lipid molecules. So it is not just a wall, it's basically a double wall. So like soldiers standing back to back and fighting to the outside and letting only through from one side to the other one should come from. Two back-to-back layers made up of 3 types of lipid molecules, the phospholipids, cholesterol, and glycolipids. Phospholipids, cholesterol, somewhere, the yellow thing, these yellow things, and glycolipids. Somewhere, the very purple ones. Okay, whatever, they're different ones. About 75% of the membrane lipids are phospholipids, lipids that contain phosphate groups. Present in small amount of cholesterol, a steroid with an attached alcohol groups are OH, and various glycolipids, lipids that have sugar carbohydrate groups attached. Okay, we're going to concentrating on these phospholipids because we seem to be way more on this illustration here, and we seem to make much more a part of this lipid bilayer. The bilayer arrangement occurs because the lipids are epithetic molecules, which means that they have both polar and non-polar parts. That is the trick. So the soldiers, if they don't shoot to both sides, they have a gun just shooting at one side. And they are not stupid, they're turning it not to itself, they're turning it just to the outside. So we're standing back to get back, and we're shooting just to the front. Just like these epithetic molecules, and on one side they are attracted to what is there, and with the other side not, and so they are arranging like that. They're epithetic. One side of them wants to be look to the outside, the other one doesn't want to. They have polar and non-polar parts. One part is attracted with like a magnet, the other part doesn't want to. In phospholipids, the polar part is the phosphate-containing head. So polar is the head. The head is polar. With a hydrophilic, the head is polar and loves water. That makes sense? It is at the outside, and at the outside is the water. Whether it's now on the inside of a cell, on the other side, on the outside is the water. So we can rearrange our little metaphor. It is a bunch of people standing in a line waiting all for a cup of water. And they have their arms to the front, that's by standing all back to back, and they're waiting to get the water. And that is obviously the outside, and that is their positive cup of water. We're going to find a word of P, some type of it, for a cup. And because like likes the non-polar parts are the two long fatty acid tails which are hydrophobic. So the head of the phospholipids wants the water, the tail not. That's their own arranging with it. Because like seeds likes the phospholipid molecules orient themselves in the bilayer with their polar heads facing outward. In this way, the polar heads face a watery fluid on either side, cytosol on the inside and extracellular fluid on the outside. There's the plasma membrane and then the cytoplasm with the fluid portion cytosol. So the bilayer is orientating all like that, and the polar head points always to the water outside of the cell and to the cytosol, the fluid part of the cytoplasm inside. In this way, the polar heads face a water. The hydrophobic fatty acid tails in each half of the bilayer point towards the center of the membrane, forming a non-polar hydrophobic region in the membrane's interior. So in the center is a non-polar and hydrophobic region. In the center is no water, and water is also not welcome there. And it's also not polar. Glycolipids account for about 5% of membrane lipids. Okay, there are phospholipids, these are the ones with the heads, the polar heads, wanting water, and then there are 5% are glycolipids. Their carbohydrate groups form a polar head. These glycolipids and various glycolipids that have sugar carbohydrate groups attached. So there are glycolipids which have a sugar carbohydrate attached. And the carbohydrate groups, so the part of them which is carbohydrate of the glycolipids is the polar head. And the tails are non-polar. So the same as with the phospholipids, and the head is consisting of whatever the phospholipids head consists, equally polar, and their tail, or whatever that exists, is hydrophobic non-polar, or at least non-polar. We don't know where it is. Glycolipids appear only in the membrane layer that faces the extracellular fluid, which is one reason the bilayer is asymmetric. Okay, these glycolipids are only at the outside. These glycolipids, they are only on the outside. They are the ones with very big bottles. They want a lot of water and they are not stupid. They know that at the outside is more water than on the inside. They are geniuses, these glycolipids. The remaining, it's easier to remember, just I think. The remaining 20% of plasma, it's asymmetric. Asymmetric because the glycolipids are only on the outside. It's actually not that bad, because the genius glycolipids are facing the outside with because we want so much water, and we know the outside is much more water. The remaining 20% of plasma membrane lipids are weakly amphiphilic cholesterol molecules. So then we have 20% left. So we have 75% phospholipids with a head, polar, wanting water. Then we're having 5% glycolipids with a carbohydrate hydrophilic head which wants also water and is also only on the outside. And then we're having the cholesterol molecules. These are also slightly amphiphilic, so one part wants water, the other not. And they're interspersed among the other lipids in both layers of the membrane. Yes, they're just interspersed among the. It's just for sense. They're just from time to time there. The tiny OH group is the only polar region of cholesterol. This tiny OH group is and it forms hydrogen bonds with the polar heads of phospholipids and glycolipids. So they're having a head, or a part of them has OH alcohol atom, and that part of the cholesterol makes a hydrogen bond with the polar heads of all these phospholipids standing there, and also the glycolipids. The stiff steroid ring and hydrocarbon tail of cholesterol are non-polar. They're having somewhere also a steroid ring rings and the hydrocarbon tail. Now on the graph it's sort of like that. Having this purple or blue head, then these 4 yellow beads, and then a little purple tail. So the head, that is the alcohol group attached, with that it can attach to the polar heads of the glycolipids and also of the glycolipids, of the phospholipids and glycolipids. Then they're having the non-polar yellow steroid rings, here yellow short, and they are fitting among the fatty acid tails of phospholipids and glycolipids. The plasma membrane typically has more cholesterol than the membranes of organelles. The plasma membrane typically has more cholesterol than the membranes of organelles. Okay. No, that was the lipid bilayer, what we said here. We understood it all. We don't have time. If you were here for cytology, you can go directly to the next part of cytology. Maybe we'll continue with that. We try to make a little presentation and save the information saved here in this little section. If you were here for that, you can go to the next part. If you stay here and participate in the 2-minute tiptoe yoga, then it's with your arms. The feet are standing together, leaving the arms up, come up with the tiptoes, out and down, and come always to the big toe with the weight in and up. Out and down. In and up. Out open to the side. In and up. Out open to the other side. In and up, make an inch space between the feet. Out and down. In and up, middle. Out, sink down, stay on tiptoes. In and up. Out, over. In and up, tiptoes. Out, down again. In and up. Out and down with the heels. In the arms up, come out on tiptoes. Out, sink down, stay on tiptoes. This can go onto the knees. In and up. Out and down. In and up. Out and down. One foot to the side, feet standing like that. In, arms up. Out, bend the front leg, come up on the tiptoes. Reach in front. Breathe again in. Out and come back. In, straighten the front leg. Out and bend it. And down, change the leg, breathe in. Out, bend the front leg, come out up onto tiptoes. In, reach in front. Out, lean back. In, straighten the front leg. Out and down again, back down, down. Then lift the arms up. And out and down. Stretch the left leg. Stretch the right leg. In middle. And out, come slowly around back, up at the top of the backward shoulder. In the arms up. Bring the hands to touch and allow them down to heart side. We don't have more time. I thank you very much and until next time.